Last speaker for this morning session, but not least, we've heard of Dr. Panda yesterday during the public lecture, so I'm going to just briefly introduce him. He's a professor at the Salk Institute in California. His research focuses on the circadian regulation of behavior, physiology, and metabolism in model organisms as well as in humans. So Dr. Panda discovered the blue light sensing cell type in the retina, and that it entrains our master circadian clocks, that it affects mood and regulates the production of the sleep hormone melatonin. Dr. Panda has received the Julie Martin Mid-Career Award in Aging Research, the Dana Foundation Award in Brain and Immune System Imaging, and was a Pew Scholar in the biomedical sciences. So without further delay, thank you so much, Dr. Satchin Panda, for being with us today.
Thank you. Okay, I'll try to be loud until... Thank you, Tammy, for inviting, and I'm really enjoying my time here. What I'll do is tell you about the publication almost 11 years ago, and it was really hard to get this publication out. It was reviewed for 4 or 5 journals before it came out. And I was so depressed that I thought, okay, so this is the last publication on time-restricted feeding, and I'll never work on it. The experiment was very simple. We took 4 groups of mice. They were all male, young male mice, and then they were given normal chow, ad libitum, normal chow, time-restricted, high-fat, ad libitum, high-fat, time-restricted. These mice were fed 8 hours every night. The reason why it was 8 hours was Christopher, who is actually one of the co-first author here. His girlfriend did not allow him to be in the lab for more than 9 hours. That's the reason behind this 16-8 craze that's going on. So, fortunately, these mice ate isocaloric diet, and that's the kilocal per mouse per day. The activity level, total activity levels are almost equivalent. The only thing is time-restricted mice are more active towards the end of their feeding cycle. This is the respiratory exchange ratio, so they do so very strong rhythm and metabolic flexibility. And this is the body weight, the high-fat ad lib, which is the classic model of diet-induced obesity. There are 11,500 papers all over the world. And if we give the same isocaloric diet in time-restricted fashion, then there is significant significant deficit in weight gain. This is not a reversal model, this is a preventive model. Normal chow, ad lib, and time-restricted, they actually don't have much impact because this NT group lost weight in the first weeks and then they remained parallel. And so that was the origin of this time-restricted feeding stuff, and that is the representative mouse. Then the next study was by Amandine, because as soon as we published it immediately, there was an 8-hour diet book and then a few other things. People said that if you eat for 8 hours, then you'll lose weight. We never showed that. So we had to do this experiment here, 26 weeks of high-fat diet, and these mice weigh almost 55 grams, put them on time-restricted feeding, they lose almost 20% of their body weight in the next 13 weeks. Although the weight loss is slow, if we take younger mice and put them here, then they actually lose much faster. So there is an age-dependent effect of time-restricted feeding, but the benefits still persist. And there are many benefits that we saw. TRF in normal chow does not change any body weight, but these mice, even to the old age, they maintain their muscle mass, which is very important for us we age, and it also increases their muscle performance. And over the last 11 years, there are multiple studies all over the world in flies, human, mice, and there are multiple benefits of time-restricted feeding. In humans particularly, we know that it improves quality of sleep and improves motor coordination in mice and flies. We don't know about humans. And then many other benefits here. Increased female reproductive lifespan. This is very interesting because dietary restriction or calorie restriction actually impairs female reproductive lifespan. Those who go on, women who go on severe caloric restriction below 1100 or 1200 kilocal, they actually become amenorrheic, whereas time-restricted feeding, at least in mice and rats, extend reproductive lifespan. and there is increased lifespan. So the question was, well, since the benefits are so much, then maybe the effect is pleiotropic and acting on multiple tissues. So Salk, when he started in the lab, there was pandemic, so he thought, okay, he could do a simple experiment, put the mice on time restricted feeding only for 7 weeks. So there is not too much weight difference, and then harvest 22 different peripheral organs and brain regions every 2 hours over 24 hours. There are 1,035 samples, RNA seq them, and this is just the meta-analysis of all the samples. 1,035 samples, 25,000 genes, you do the matrix, and there are essentially these 27 clusters of genes that actually change between ad-lib and TRF. What is interesting is if you project that UMAP on different tissues, then what you'll find is, for example, here in IWAT, this particular cluster is actually upregulated in TRF, but in EWAT, the same cluster is downregulated in TRF. So when somebody says an intervention changes or upregulates this gene, you got to ask in which organ, because there is an organ-specific effect, directionality of this change. But anyways, if you want to take away some top-level message, then actually the biggest impact of time-restricted feeding is on adipose tissues, followed by digestive organs, but surprisingly not on the ileum, but only on duodenum and jejunum. And then comes muscle and heart. Liver is actually way down there. So endocrine organs, for example, adrenal and hypothalamus are the next. And we found that nearly 70% of genes, or 80% of protein coding genes, are differentially expressed up or down regulated, because we just took, irrespective of time points, we just treated them as replicates. So again, there is sufficient power to believe in these conclusions. Then if we ask what are the up or down regulated genes, then it was pretty simple and straightforward. So these are the upregulated genes essentially ensure quality control of RNA and protein, and also breakdown of fat and cellular waste or autophagy, whereas downregulated genes are mostly inflammation and reduced oxidative stress in 5 or more tissues. Then since we did time series analysis, if we look at 24 hours rhythmicity, then nearly 40% of genes were rhythmic in ad-lib fed mice in at least one tissue, which is very similar to what John Hoganes in his famous paper showed. 40% of genes are cyclic. But actually, if you put the mice on time restricted feeding, that goes up to 65%. But here we're sampling only 22 different tissues. And yesterday I showed the baboon data where we have 64 different tissues, and same, 2 hours sampling and 8 hours time restricted feeding. And in those cases, the number actually jumps to 85% of protein coding genes. And then if we stack them up, the peak phase of expression of all these genes, then what we find is there is a big burst of activity in the middle of the fasting. So in anticipation of feeding, these genes begin to decline. So this is the anticipatory function of circadian rhythm. And then these are the feeding induced genes. And if we just look at what are the genes that are in the fasting phase or feeding phase, then clearly the fasting phase drives the upregulation of autophagy, DNA repair, cell cycle, fatty acid oxidation, and then again the quality control is always in the feeding phase. So this is very important, because we always think, even in calorie restriction, people say calorie restriction improves protein folding and all that stuff. It's not during the calorie restriction or fasting phase, it's refeeding phase. That's when those genes are upregulated. Now that we have tissue level transcriptome in multiple tissues, we can ask very simple question. How is fat absorbed from the intestine? How it is transported to the liver or in the periphery? How this is lipoprotein lipase breaks it down and then recycles. And the bottom line is this kind of data set gives us hypothesis to test. So the bottom line is all the gene expression signature told us that, well, time restricted feeding might have a beneficial effect in preventing atherosclerosis or reducing VLDL production or VLDL secretion. So then we tested this in LDL receptor knockout mice. Almost all, except one that came out last year, almost all drugs to reduce atherosclerotic risk depends on the functional LDL receptor. And in fact, familial hypercholesterolemia, which affects almost 1 in 150 or 200 people, that also relates to 95% of familial hypercholesterolemia is due to mutation in LDL receptor knockout mice. So this is something that will not respond sufficiently to statins. So we put the LDL receptor knockout mice on high fat, high cholesterol diet on ad libitum or time restricted. They eat the same number of calories, but the high fat, high cholesterol time restricted, sorry for all these abbreviations, they gain less weight. And then if we look at the serum triglycerides, serum cholesterol, they all reduce. And when you look at the plasma lipoprotein profile, what is interesting is the reduction in those lipoprotein is mostly in the VLDL, chylomicron remnants, IDL, and LDL. There is no change in HDL fraction in these mice. And this is something that we have also seen in humans. In humans, time restricted feeding does not bust the HDL. It usually reduces VLDL particle size, but reduces VLDL particle numbers and ApoB and all these that are reflected here. And then if we look at the plaque size, we actually see almost 30% reduction in plaque in this LDL receptor knockout mice, and these are all the quantification. Then what is the mechanism? Is the absorption reduced, or LPL, lipoprotein lipase function changes, or liver reuptake of this TRL remnants happens, or VLDL secretion? Which step is affected by time restricted feeding? What we found is the triglyceride absorption is very similar between LDL receptor ad liver TRF mice, and then the LPL activity is also very similar between those two. The clearance, hepatic clearance of this TRL is also very similar between ad liver and TRF. The only thing that changes is VLDL secretion. So the production of VLDL from liver, that's actually significantly reduced in LDL receptor knockout mice subject to time restricted feeding. And I'll get to the ApoE knockout. Since ApoE knockout mice have a defect in clearance, they cannot actually reabsorb all of the TRL remnants. They do have less amount of TG to begin with, and then of course there is also significant reduction in VLDL production. And now we ask, well, if that is so, then if we put the ApoE knockout mice, then what happens? So the ApoE knockout mice, they also eat the same number of calories. There is not much weight change, and there is not much change in VLDL, IDL, or chylomicron remnants. The serum triglycerides, serum cholesterol also don't change. And in fact, and as you would expect, there is also no change in atherosclerotic plaque. So this was a preventative model. Then the question is, if the LDL receptor knockout mice already are on high fat, high cholesterol diet, and then you put them on time restricted feeding, then what happens? So this is when we fatten up the mice and put them on high fat, high cholesterol diet, and then divide them into 4 different groups. One continues on the same diet, and then 2 groups are given actually normal chow, because this is the standard of care. People will ask, okay, so reduce your cholesterol intake, eat more healthy food. And then another group was the same diet, but they are actually in time restricted feeds. So LDL receptor knockout, high fat, high cholesterol, time restricted feeding group. And all these 4 groups, these 3 intervention groups actually reduce their fat mass almost to equivalent extent, 9 to 10% fat. And if we look at the triglyceride levels, we know that the triglyceride levels can change between fed and fasted state. So that's why we measure them in fed and fasted state. The fasted state triglycerides are very similar between normal chow, ad libitum, normal chow time restricted, or high fat time restricted. And then, as you'd expect, the high fat, let's go to the next one. And then this is the plasma cholesterol level, again, fed and fasted. What is interesting is the LDL receptor or high fat, high cholesterol fed mice, during the feeding phase, they have pretty high level of cholesterol, but that overnight fasting of 14, 15 hours actually almost reduces the cholesterol level by half, almost 50% reduction. And so this is very powerful in reducing that cholesterol. And then the question is, what is this extra cholesterol going? So we know that the bile acids are the degradation product of cholesterol, and bile acids can be excreted in stool. So there is significant increase in stool clearance of total bile acid and stool clearance of total cholesterol. And again, we did see significant reduction in plaque. And with that, I'll stop, and I'm happy to take a few questions. Thank you.
Brian first.
Really, really great talk, really interesting. I confess to not reading the paper that you were both talking about, the work you're talking about. Did you look at immune cells and how those were affected by time-restricted feeding? There's a prediction that macrophages may also be affected since they contribute to atherosclerotic plaques.
So we didn't actually look specifically at different cell types, immune cell types, and then we thought that, okay, if you take the spleen, then it's so heterogeneous that it's, unless you do deconvolution, it will be really hard. So the next step is to figure that out. Coming back to macrophage, actually we did macrophage efflux assay, and these are very difficult assays, but we didn't see any difference between time restricted and early. And then the same thing again in patient samples also, we don't see much, but at the same time I must confess that we are not taking really patients who have pretty blown up atherosclerotic risk.
Excellent talk. Of course, I have a clinical question. You mentioned briefly time-restricted feeding in humans. What do you know about that? What studies have been done so far? Is that something you have seen in healthy volunteers, or just like small sample studies, or is there something more? Because I was looking at this, I didn't find much.
Yeah, so we just completed a randomized controlled trial sufficiently powered on 110 patients with metabolic syndrome. So they met at least 3 out of 5 criteria, and that paper is under review, so you hopefully will see it. But the firefighter study was, mixed population, and there what we found was those who started with higher level of, say, blood glucose, cholesterol, or blood pressure, they actually improved. But again, the subset was very small, 10 to 15 or 20 with each condition. So you'll see soon the metabolic syndrome study.
I'm wondering how quickly the benefits of time-restricted feeding can be realized in the mouse models, but just thinking about humans. So, for example, instead of having them on time-restricted feeding for many weeks at a time, maybe a week on or a few days a week, a week off, just thinking about people not being able to stick with the program or willing to come back to it.
I always say that, if sleep is important, can people just sleep for 5 days? So, bottom line is, it should be a lifestyle. That's why we say 10 hours time-restricted feeding is something that many people can do every single day. And although in mice we have done 5 days of time-restricted feeding and 2 days of ad libitum, we do see some benefits up to 12 weeks. We don't know what will happen to those mice if we continue for 6 months or a year. And in humans, again, what we are seeing in many studies, if the patients comply for 5 to 6 days in a week, we still see a lot of benefits. And then the question is how quickly you see benefits. It depends on what benefit. For example, the patients will say that in 1 or 2 weeks, there is no randomized controlled trial on this, but acid reflux or heartburn goes down. We know that in the US, 68 million prescriptions are written every year. This is prescription. Non-prescription, there are a lot more. We're taking acid reflux medication. So that improves in 2 weeks. 3 to 4 weeks, people will report that their sleep improved significantly, so that's why they stick with it. We haven't looked at LDL cholesterol and other stuff, but I think it will take a long time. When you put a CGM, of course, the nighttime glucose reduces because they're not eating. So in that way, you can argue that the 24 hours average also reduces within a week or 2.
One last question, Ray?
Have you looked at the effects of alcohol consumption?
We actually see time-restricted feeding significantly reduces alcohol consumption because there is not too much time to drink. And this was very important for the firefighter study because firefighter, They do have active program for alcohol reduction and the struggle, and then we found that no, they just stop eating and drinking at 8 o'clock in the evening. They can significantly reduce alcohol consumption.
Please join me in thanking everyone.